Good morning, good afternoon, good evening, depending on your time zone and when you watch. Thank you all for coming to our Locust Walk RNA Innovation Conference, hosted in partnership with Dr. Frank Slack, Director of the Harvard Medical School Initiative for RNA Medicine, and Dr. Phil Zamore, Chair of the UMass Chan RNA Therapeutics Institute. This is our second Next Gen Therapeutics Conference. The focus of our 2022 conference last spring was stem cell therapeutics in partnership with the Harvard Stem Cell Institute. We're thrilled to put on what we believe is a first-of-its-kind event that combines the best of an investment banking conference with investors, a partnering conference with strategic partners, an academic conference where cutting-edge research is presented. For this conference, we have 27 companies presenting and 6 academic scientists sharing their innovations that are either in the process of becoming a new company or have the potential to become one within the next 12 months. The goal is to find novel next-generation deal flow for investors and partners and just those interested in the space. More than 550 of you, the investors and strategic partners and academic innovators, have signed up for the conference, and we are appreciative of you spending your time with us. So, as I said, the goal is to find novel deal flow and innovation for people interested in investing or partnering in the space. Additionally, we have assembled a world-class list of experts for our keynote, our lunchtime, and our afternoon panel discussions. So, just as a brief commercial and background for those of you who are not acquainted with Locust Walk, we're a global investment bank that integrates corporate development strategy and execution with capital raising for innovative life science companies. We help companies where a deal is either an input or an output from a strategy. We then assist on actually executing those strategies on a global deal or regional deal basis, helping on both the buy side, easy for me to say, and the sell side. We also help companies raise private capital. We do this on a global basis. We have 40 people on the team, and we have 5 offices in all the major biotech hubs. So before diving in, I also wanted to acknowledge and I'm very grateful for the Locust Walk team who worked tirelessly to put this event together. Thank you for putting on such a fantastic event with so many great speakers, companies, and of course, attendees. For this first segment, we have 40 minutes allotted for this keynote discussion. We will be recording this session as well as the lunchtime and the afternoon panels, and a playback will be available shortly afterwards for attendees through the same MeetMax system. So without any further ado, I am now pleased to introduce Dr. Frank Slack, Director of the Harvard Medical School Initiative for RNA Medicine. Dr. Slack is the Shields Warren Mallinckrodt Professor, Department of Pathology, and Director of the Institute for RNA Medicine at Beth Israel Deaconess Medical Center. The Slack Laboratory is at the forefront of the small molecule, small RNA revolution. They co-discovered the first human microRNA, LET7, and showed that it is a tumor suppressor that controls key cancer genes such as RAS, SMIC, and Lin28. They are developing LET7 and a second microRNA, miR34, as novel cancer therapeutics, with miR34 already in phase 1 clinical trials. They also proved that the microRNAs act as key oncogenes and develop strategies to target these oncomers for cancer therapy. The research also extends to discovery of additional novel RNAs in development for cancer, aging, and diabetes, and minor conditions, as well as identifying novel SNPs in the non-coding portions of the genome with an eye to identifying the next generation of actionable targets in cancer. Welcome, Frank. Dr. Phil Zamore, the Gretchen Stone Cook Professor of Biomedical Sciences, Chair of the UMass Chan RNA Therapeutics Institute. Dr. Phil Zamore has been an investigator of the Howard Hughes Medical Institute since 2008. He is chair of the RNA Therapeutics Institute, which was established at the University of Massachusetts Medical School in 2009. He is also the co-founder of a tiny company called Alnylam Pharmaceuticals and also Voyager Therapeutics. Maybe you've heard of those two. Dr. Zamore's laboratory studies small RNA silencing pathways in eukaryotes and prokaryotes, including the RNA interference, RNAi, microRNA, Peewee interacting RNA pathways. Dr. Zamore and his collaborators seek to use these insights to design novel therapeutics for human diseases. Under Dr. Zamore's mentorship, the Zamore Lab has produced dozens of researchers working at the top institutions in the United States and abroad. So welcome, Dr. Frank Slack and Dr. Phil Zamore. Thank you for joining.
Thank you.
Okay, we're going to get right into it. The spirit of this is to get the academic perspective on some of the innovations that have transpired in the RNA space. So there's no topic that's off limits for you to discuss. You can regulate as you need for yourself. So maybe perhaps, since it is so unique to have RNA institutes, and there's two of them actually in a relatively close geographic setting, I'd love to hear from each of you. What was it like to give us a background, first of all, on your institutes? What are the goals? But how did you each come about starting this and pioneering this type of innovation? So maybe Dr. Slack, if you want to give it a shot first, and then we can talk to Dr. Zamore next.
All right, sure. Well, thanks, Jeff, and thanks everybody for tuning in. So for me, the sort of vision began in the sort of mid-2000s when we realized the awesome potential of RNA as novel diagnostics and therapeutics. I shouldn't say we, I mean, but I mean the field. There were, of course, some fantastic companies already starting in this space that were really proving that these technologies could allow us to target almost any RNA in the human disease space. And so at the time I was at Yale University, which had an outstanding group of basic RNA researchers. And so I teamed up with some of the sort of luminaries that are at Yale, including John Stites and Ron Breaker, and we started the Yale Center for RNA Science and Medicine. And we were very successful in bringing the Yale community together and thinking about how to translate their amazing RNA discoveries into new potential medicines. And that activity seems to have attracted the attention of Harvard. And in 2014, I was recruited to Harvard Medical School to start an institute of a similar nature. In particular, I was based in the cancer center and in the pathology department at the IDMC, who had recognized the sort of value in these new classes of RNAs as both novel diagnostics, potentially, but also as novel targets for therapy. And so what we try to do at the HIRM is bring together the community. We realize now as a community that many of the same technologies and the same ideas are sort of applicable across different medical modalities, and more than that is bring people of different expertise together to try and solve some of the big barriers that are in our field. Barriers like, for example, efficacy, targetability, delivery, that sort of thing. And even more than that is bring in people who haven't worked in the RNA medicine space into this field. In particular, expertise in biomedical engineering and computational biology in clinical sciences to allow these innovations to make their way into the clinic as quickly as possible. And part of that is to try and encourage translational science amongst our faculty. One way that we do that is we try to encourage our faculty to patent, license, and sometimes spin out their companies to bring these sort of innovations to patients as quickly as possible. So that's how we started, and that's the goals. We're currently at about 60 faculty that work on all different areas of RNA medicine. We're agnostic to the type of RNA that's being used. We're agnostic to the way that RNA is being targeted, to the disease that's being targeted. Really, the idea is just to form a large umbrella, a big boat, and all go along for this fantastic ride. Sorry, Jeff, I think you muted.
I got my one mulligan. Can't do that again. You only get one per webinar. So just before going to Phil, so this is a pan-Harvard thing. This is something that there's people in all the different hospitals. How do you span so many different institutes to get people to collaborate? And I guess, is there any more you could share about the size and scope and how you tame that?
Yeah, so I mean, it's one of the advantages of being in the Harvard system is that it's so large and so many fantastic researchers. It's also one of the challenges to sort of bring everybody together. I can tell you that we're not in all sitting in one large building. This is sort of a matrix institute. where the faculty are spread across 11 different hospitals and research institutes. And recently, just we now have joined forces with MIT as well. So it's now across Harvard and MIT. The goal is really to bring people together at events like our seminars, our symposia, our workshops, our research in progress. We have some pilot funds that we use to bring people together to work on projects. So it's sort of a community that's coming together to try and tackle some of these big issues in the field.
Got it. Cool. Thank you. Dr. Zamore, over to you.
So the original concept for having an RNA Therapeutics Institute came from Craig Mello right after he won the Nobel Prize with Andy Fire for the discovery of RNAi. And his original concept was that the university would build a new building and use that building to start a series of novel programs for the campus, one of which was the RNA Therapeutics Institute. And initially, there were 4 of us who co-directed the institute. Victor Ambrose, who discovered microRNAs with Gary Ruvkin, Melissa Moore, who ultimately left UMass after 13 years to become CSO of Moderna, myself, and Craig. And from the very beginning, the concept was to bring fundamental RNA science and translational research together in the same space. It's a formula that's worked well across our campus. Physical proximity is the best predictor of impactful collaborations. Literally, we are trying to build a framework physically in which people bump into each other, so that people who study RNA delivery, like my colleague Anastasia Korova, who came to the RTI out of decades of industrial experience, and my colleague Craig Mello, who has worked exclusively in C. elegans, they are collaborating based on a shared set of interests and love of RNA. And out of their long-term collaboration came, for example, Atalanta, the CNS siRNA company that was co-founded by 3 faculty at the RTI. We're small. There are only 12 of us. Our plan is to grow to about 16 and continue to balance hiring fundamental researchers and translational researchers. We also are one of the few RNA therapeutics or RNA centers that can prepare enough synthetic RNA for a complete preclinical tox package in non-human primates, even on our own campus. I once had Anastasia rush into my office and tell me to hold a vial while her postdoc stood there and photographed me. And I kept saying, "What am I doing?" And she said, "We'll tell you later." And it turned out I was holding the first 5-gram batch of siRNAs produced on campus. So the Institute really values community, and we've tried to spread that message through our annual symposium, through our outreach programs to the Worcester Public Schools, through our coloring book for small children on mRNA vaccines, and by spinning out companies that develop novel RNA therapeutics for unmet needs.
Very cool. Different approaches, but obviously both have been successful. So I'm going to put myself in the shoes of somebody in the audience. I'm at an investment firm or I'm at a pharma company. What is the best way, and I'll start with you, Phil, to access innovation at your institute? Everyone talks about roaming the halls and knocking on doors and talking to people in the lab, but is there any kind of formal structure, or is there a way that you've tried to cultivate those connections? Maybe you could talk a little bit more about how that has been processed.
So I think the best way is to come to our annual symposium. The symposium brings together about 350 people on campus with another several hundred watching online. And virtually all of the graduate students and postdocs present their research either at posters or at short talks. So it's an opportunity to talk to the actual people who are doing the work, to see the breadth and depth of the work coming out of the department. Our tech transfer office attends the conference, as do many of our close partners in industry. And plus, we have great speakers. This year, Caitlin Carico is our keynote speaker. So that's probably the easiest way to learn about what we're doing. We always welcome people visiting the department. We can have you come and spend a day talking to faculty, talking to the tech transfer office, meeting with students over lunch. So there are a number of opportunities. And then, of course, I think most people know about the work our faculty are doing in CRISPR, in RNAi, in mRNA therapeutics, circular RNAs. So there are a number of ways.
So to give you a 20-second commercial, when is your next symposium if people on the audience want to come?
It is June 20, yikes.
It's coming up.
It's on our website, the end of June.
Go to your website and sounds like a great opportunity to learn more. So Frank, maybe you could share how to best interact. I know there's more faculty and some are more entrepreneurial minded than others. How do you think about navigating the Harvard system for RNA innovation?
Right. So, I mean, I would say similarly to the way that Phil just mentioned, but we have an additional avenue, if you will. The HIRM has a business advisory board that meets twice a year, or it could be meeting more frequently now that we have more activity, where our faculty have a chance to actually speak about their new science to a group of VCs and RNA entrepreneurs. So if anybody's interested in learning more about our business advisory board, please, you're welcome to send me an email. If you'd like an introduction to any of our faculty, you're also welcome to send me an email, and I can try to reach out on their behalf. Because we're a matrix institute, each of the 11 institutions that's associated with the HIRM has their own tech ventures office, and we would have to put you in touch with those individual officers. So it's not a centralized process, but we try to centralize the access to information. But then once somebody wants a deeper dive, we'd have to hand you off to the individual offices.
Got it. Nope, that makes sense. And just go for it.
June 21st through 23rd.
Very quick thinking. Good stuff. Thank you both. So I guess, just to follow up from that, you've talked a little bit about some of the innovations that have come out of both of your institutes. It would be great to hear some of the stories or companies that you're particularly proud of or interested in, either you had a hand in creating or some of your colleagues did. Give a flavor for some of the innovations that have come out. And then in follow-up, the next set of questions will be about, well, what's next? So maybe I'll start with you, Phil, about what's come out either from your lab or from the institute that you're particularly fond of or interested in.
From my lab, in the years since the RTI was founded, my colleague Guangping Gao, who leads the Gene Therapy Center, which is similar to the RTI in that it brings together fundamental and translational research in that space. He and I co-founded Voyager, and the work in my lab that ended up in Voyager was the concept of artificial pre-microRNAs, which can be engineered into AAV vectors to generate siRNAs in vivo. So durable viral vector delivery of RNAi, essentially. From my colleagues, Craig Mello is a co-founder of Editas, and he and Anastasia and Neil Aronin co-founded Atalanta, which uses Anastasia's technology of dye siRNAs that, when injected into the CNS by a variety of routes, produce extraordinary coverage of the brain, many cell types, and and really makes it possible for the first time to tackle devastating neurodegenerative diseases using infrequently delivered RNA drug. My colleague Eric Sonheimer co-founded Intellia, and he and my other colleague Wen Xu continue to collaborate, innovating novel alternatives or adaptations of prime editing, yeah, I think that's a good representation. I'll let Frank take over.
It's a good list. Go for it, Frank.
Yeah, so I would say one of the proudest things that I think we've done at the HRM through our business advisory board is that we've launched at least one company a year directly through sort of activity that has come from basic science discoveries in our institute. I'll just give. Well, I mean, there are multiple examples, but I can give one from my own experience, and that was a company that I founded with Richard Gregory to try to target RNA binding proteins, in particular the RNA binding proteins that are regulating microRNAs. That company started out as 28-7, named because of the original RNA binding protein we were trying to target called N28, and what it regulates called that7, but is now called Redona Therapeutics. That company is, almost ready for IND studies, IND enabling studies, and may try to go public in the next few years. So very excited about that potential. I would say some of the other exciting developments are companies launching around RNA modifying enzymes. We're learning fast that these RNA modifying enzymes show up in disease. Often they're amplified or they're mutated in disease, and targeting them is, while it's not trivial, is still possible. And then other sort of areas of sort of novel expansion into novel areas include new types of RNA modifications that are found in RNA that could be useful in the next generation of oligonucleotide drugs. So I think it's a very exciting time generally for these sort of innovations. For me, I'm extremely happy whenever I do see one of our basic science faculty, think more carefully about how their invention could be applied. It's one of our goals to try to drive that innovation into an application. And when that comes out as a new company, that's often one of the surest ways that we're going to see that technology make its way into the clinic. So we're very, very focused. Well, that's sort of focused, but one of our focus areas is to try and bring these innovations to investors to potentially bring them into the clinic.
Yeah, that's definitely unique among, frankly, the two of you and your two centers, because a lot of academics have an interest in publishing, which is obviously still important, but the translatability into an investable company or a partnerable asset is harder to come by. And so it's clear that both of you have developed cultures that have encouraged this and views that as the goal, not just as an ancillary benefit of it, which is, I think you both acknowledge is where it gets closest to patients. So before going over to you, Phil, to get your views on what's coming that interests you, maybe, Frank, if you could speak a little bit more about the RNA targeting enzymes and some of these new RNA modifications. Obviously, I'm sure there's confidentiality involved in some of this, but just at a high level, what is different about these approaches compared to what's been done before, and how should we think about this innovation?
Yeah. I sort of hesitate to bring more complexity into the field of sort of RNA biology, but we're still grappling with the fact that there are thousands of RNA species that are being made within our cells. Maybe tens more even groups of RNAs than we actually appreciated just sort of 2 decades ago, and now we have to grapple with the fact that these RNAs are modified in over 100 different ways. So before I tell you, answer your question, let me just tell you that I think that this complexity, while it is daunting, is opening up a lot of opportunity that we should be able to exploit for better diagnostics and therapeutics. I mean, RNA modifications that were discovered almost at the same time as RNA was discovered back in the 1950s and 60s. RNA is modified in 170 different ways. Nucleotides can be, modified in the ways that you're all familiar with at the 2' position of the sugar, but there are 150 different modifications that viruses, bacteria, even our RNAs use, and they've been pivotal in the success of RNA medicine. I mean, the 2' modifications are probably the most important modifications that have really made RNA medicine a reality. And then, of course, pseudouridine really made mRNA technologies usable in the clinic. So these modifications, they weren't, they were in nature. So now we just need to learn how to sort of capitalize on these other 167 that we haven't yet really worked on that much. So that's the first part. The second part is that there are enzymes that are often involved in making these modifications. And as we dig deeper into looking at the modifications, we realize, that these modifications can be enriched in certain disease states. And then the enzymes themselves show up as being enriched in these disease states. And because they're enzymes, they can be targeted with small molecules and other approaches that block active sites. And so there are companies that are launching in this space already. You've probably heard of some of the methylases that are already close to entering clinical trials, like Metal-1, Metal-3, that are being developed by companies like Storm Therapeutics. And then, of course, there are other enzymes like the ADARs, which are converting an adenosine residue into a residue that can base pair with a different partner. It changes the coding potential of some mRNAs, but also changes the ability of those mRNAs to base pair with other RNAs. Those enzymes are not only being used, to make modifications that are maybe going to be useful in therapeutically altering the sequence that's found in a particular RNA, but also those enzymes are showing up as being important for certain diseases as well and can be targeted. So that's I think a pretty exciting area of research going forward.
Well, Phil, we didn't leave much ground for you to cover here. So if there's anything that you've seen that's an innovation in RNA that excites you, and you can feel free to amplify anything that Frank said, but if there's other areas of interest, we'll love to hear about it.
Well, I think the biggest lesson of CRISPR-Cas9 is that the historical fear of using foreign proteins as intracellular therapeutics is probably overblown. So I think many of us, myself included, were worried that if you put a bacterial protein into a eukaryotic cell and expressed it for a considerable length of time, you'd end up with an immune response that would actually clear the cells that you had transduced. I think we now realize that that's not the case, and that there are reliable, safe strategies for introducing a host of proteins that are not normally found in eukaryotic cells, and using them in the context of RNA therapeutics. Things like editing enzymes, things like reverse transcriptases, and so forth, that are using RNA as a template, for example, to correct a defect either in the genome or in the RNA.
Interesting. So before going on to the next related topic, I thought, Phil, since I know your research wasn't yet at a spot where you could disclose anything non-confidential, I think Frank is going to be presenting later today. If there's anything you're open to sharing just non-confidentially about some of the things you're working on, that'd be great to hear.
Well, I think we still don't really have a sense of the full potential of RNAi, and our lab continues to use a combination of biochemistry and genetics to really understand if there are better triggers than siRNAs to induce RNAi, and how we can improve upon the delivery of siRNAs by innovating perhaps smaller siRNAs. So that's an area I'm very actively interested in. And then I would say in general, Frank alluded to RNA modifications. I think we have a very rudimentary understanding of the effect of RNA modifications on RNA control elements and their effect on translation. And I have colleagues in the RTI who are actively pursuing those studies.
Well, look forward to when you're finally able to share it with the world. So I guess we'll all have to stay tuned. One thing we're going to talk more about this in the financing panel, so in the partnering panel, so I'm not going to cover market conditions here, but what's interesting to me is that the last few years saw a whole bunch of novel cell therapies and gene therapies from, iPS derived or AAV or any type of viral ex vivo, in vivo. And we're seeing the difficulty of those types of modalities in part because of the challenge to manufacture, the cost to manufacture, process development. In many ways, these are actually processes that are masking as therapeutics. They are therapeutics, but they are really just processes. And so RNA has definitely come on and not had some of that same baggage or overhang. We're seeing more investment and interest in the RNA space than some of the traditional cell and gene, where I think there's what I've termed indigestion from a lot of the folks watching this webinar. And so I guess since you both had exposure to both, I kind of threw out one hypothesis, but can you share your views as to why you think there was either overinvestment in one and now RNA is taking its place in the sun, or is this just a natural evolution to edit the RNA as opposed to the genome or replace the, give us some context for how we should think about the evolution of these next gen modalities.
So I don't think there's been overinvestment in things like AAV. I think that there actually has been underinvestment in the contract manufacturing space. It's not that AAV manufacturing is a problem in search of a massive solution. It's that it's more like car manufacturing than small molecule manufacturing. It's got a lot of moving pieces. The assembly line has to work perfectly, and academic AAV synthesis has been fairly successful for preclinical trials, but there are not enough contract manufacturers to meet that demand. I think in contrast, RNA therapeutics that are synthetic RNA can slot right into existing chemical manufacturing specialists. And I think it's also a lot more straightforward to build synthesis facilities in-house for companies. But I think in cases where the biologically based synthesis of RNA therapeutics has been necessary, like for mRNA vaccines, it's the simplicity of the process that has made it so scalable. There are some differences between what Moderna does and what a postdoc in my lab will do to make a long mRNA, but it's not a lot. And, given enough money to buy enzymes, a company can set up mRNA manufacturing much more simply than something like AAV.
Maybe I can just add to that. So recognizing the things that you just talked about, Jeff, Harvard and MIT came together to build an academic cell and biologics manufacturing facility. Now, very, very rare. I mean, actually, UMass has something similar that maybe Phil wants to talk about in terms of manufacturing RNA therapeutics. But this was sort of really designed to manufacture cell-based, for example, new innovations in CAR T therapies, immunologics, and because I was on the committee that launched this, I was a strong advocate for RNA as well. And so there are clean rooms for manufacturing RNA right here on the Harvard campus. So it's possible even for academics to make those first preclinical talks and maybe sort of even getting to the point of IND enabling studies on campus before you even have to go to a CRO. But I would agree with Phil. I mean, I think RNA is sort of shining at the moment because of the success of companies like Alnylam and Ionis in getting products to market. And there are patients now that are benefiting from these types of therapies, and we recognize that it can be done. So I think some of the negativity around RNA that existed in the first generation drugs, I think that's worn off, and innovation has won over, and so I believe that there are a number of reasons why RNA is now having its moment. It's probably many different areas, but I think success builds success.
Yeah, it's proven at this point on multiple different modalities. So we only have about 3 minutes, so maybe 90 seconds each. And I'm curious because one of the hottest topics right now is AI and ML. And I'm curious, just based on what you've seen at your institutes, how that evolution in technology might be applicable or impact the RNA space. Maybe Phil, if you want to start.
Yeah, well, my lab uses machine learning to understand the pRNA pathway. And so that's a classic academic use of the tool. In terms of translation and making drugs, I think ultimately matching disease and transcription profiles to existing drugs will really be the place where AI will shine. I also think there's a real challenge that hasn't been met, but will be transformative when it is met, for AI to solve the three-dimensional structures of RNA. Right now, what AlphaFold does for proteins is not available for RNA. It still requires user intervention.
I would say from my point of view, I mean, some of the proudest moments that I've had are sort of bringing computational biologists with those sorts of skills into this space. Mainly, my thinking was just to help us sort out all of the transcriptomics that's coming out of different large programs. Sorting out not only just the bulk RNA sequencing that's happening, but what about the single cell and now spatial sequencing? That's really where I think it's going to shine. We already know that RNAs can make their way into diagnostic tests that can be prescribed for patients, but we're just awaiting an explosion in the sort of RNA diagnostics are going to come from sort of pathologists staining slides, with RNA markers and getting diagnoses from that. But sorting that out with 100,000 RNA species being expressed in our cells is a challenge. That's
cool.
What it's going to show.
Spoken like a true pathologist, but awesome. Well, thank you both. Really appreciate your insights, your expertise, and sharing that with the group. We're out of time now for this panel, but we will have presentations beginning in about 1 minute. They'll go until noon. And at that time, we'll have a discussion about the future of RNA targeting small molecules. The presentations will resume at 1 p.m. and go until 3:20, all East Coast times. Apologies for not saying that earlier. And we'll have a conclusion to the conference with a panel discussion on RNA partnerships, M&A, and financings. So thank you all and look forward to a great day.
Bye-bye.
Everybody.